everyone. Welcome to Chapter 4. Now, in Chapter 4, what we're going to be dealing with is carbon. We're going to be talking about carbon chemistry and getting an idea of the basics that we're going to need to have to understand the next chapter, Chapter 5, which deals with the complex large organic compounds that we use to make up cells. Now, what is important about this is the last chapter before our exam, chapters 1, 2, 3, and 4, exam 1. Now, I'll talk a little bit about that at the end, just reminding you that you have an exam after this. We'll discuss that in an email more than anything. But what I want to do is take a look at this chapter, carbon and the molecular diversity of life. Just reminding you, if you want something you've never had, you must be willing to do something you've never done. So let's take a look at carbon. Now, one of the big things we're going to talk about is how carbon can form large compounds, things like that. Now, I will say this, that 2, 3, and 4 are going to be my big things for here, okay? That's going to be my big units for this. This will be the meat and potatoes for this. The first objective is a little bit harder to really kind of discuss a little bit, but I'll talk about it. Now, what I want to discuss here is the idea that carbon can form the backbone of life, because most of your molecules that we use in living systems are based heavily on carbon. Carbon can form large compounds that are quite complex in structure with a wide variety of shapes, structures, and functionalities. Things like proteins, DNA, the carbohydrates, to name just a few of them, are so crucial and critical to life, in fact, that it seems to be almost necessary for it, is these carbon-based compounds. Now, one of the things I like to say here is that carbon, it has friends it likes to bond to. Now, I always call these the usual suspects, chomps, C-H-O-N-S-P. Carbon loves to bond to carbon, so it bonds to itself. Carbon loves to bond to hydrogen. Carbon loves to bond to oxygen, nitrogen, sulfur, and phosphorus. Okay, chomps, C-H-O-N-S-P. That's a good way of remembering the things that carbon loves to bond to form these compounds. Now, first off, I always mention this. This is an example that's given out there a lot, very well discussed in the world of biology. And evolution in itself is this really fascinating area here, which was helping to try to understand the origin of life. And Stanley Miller's experiment back in the 50s really begun to show this when he was a graduate student and conducted this research, is that he made a model atmosphere inside of chemistry equipment and was able to collect samples from that and found that molecules that are essential for life could be produced, what we would say, without living things. This kind of got rid, really the big thing that we could say is this got rid of the idea of vitalism, that these things had to be made by living things only. That it showed the idea that living molecules that are found in living things, that's how I'm trying to say this. Molecules that were only thought to be in living things are now also known to be made by natural phenomena. And what we began to see is that perhaps this led us to the idea that abiotic synthesis of life may have occurred. And we do know that actually that if the atmosphere, what we know now about early Earth's atmosphere, if Stanley Miller's experiment contained that exact composition, very well may have exploded. So, in fact, though, many experiments have been done in this world of experimentation that have shown that things like ammonia and hydrogens and things like that are precursors to make molecules like amino acids and very more other kinds of compounds out there that are needed for life can be created by natural means through a slew of a variety of energy sources. which possibly points towards the idea of life coming from non-life. It does not prove it by any means definitively, and I do want you to know that. It might be that, I'm going to kind of make a point that I completely accept evolution as a scientific fact, and now I don't let my beliefs out there very much, but I will say this is that whatever my beliefs would be, if my beliefs were in a very naturalistic, let's say it was a very naturalistic worldview, this would not be the most accurate way of explaining that. Okay, but it is a classical example of that molecules for life, molecules that we see with life, are absolutely capable of being created naturally through almost any kind of energy source that we have seen helps to do it. Now, that takes us away from the old evolutionary biology thing. It gets us back into the meat and taters, as we like to say in the South, the stuff you're going to get filled on. And this is the stuff that's going to be the bulk of your meal. And starting off is the difference between organic and inorganic compounds. Now, one of the things I always like to start off with is organic compounds must have carbon and hydrogen. That's it. It must have carbon. It must have hydrogen. CH4 is the simplest organic compound you can have. Okay, you can't have anything more simple as an organic compound than CH4, methane. Now, what about C6H12O6? Okay, it's got carbon, it's got hydrogen, but it's also got oxygen. Now, remember CHOMPs, CHONSP. Those are the molecule chomps. Hard to say chomps. It's hard to say it, but chomps. And CHONSP or CHOMPS, those atoms love to also bond to carbon. So these are common. Now, it also may be other things out there. I don't want you guys to think that. Those of you right now are currently taking organic. You may also begin to see that in organic chemistry, there's things like alkyl halides that happen to have some of the things like chlorine or bromine or something on it. Okay, now the halogens. Now, what I want to talk about is that they must have carbon, they must have hydrogen, but they also may have the other things like oxygen, nitrogen, sulfur, all that. Inorganic compounds, they have a bunch of things. They can have carbon or they can have hydrogen. They don't have both. Things like sodium chloride, water, H2O, carbon dioxide, CO2. These things, now they may have calcium, iron, aluminum. Now, great example of this. Great example, a very important organic compound called hemoglobin or myoglobin, also has iron in it. Okay, so I do want to make sure that you understand there are exceptions to everything. Okay, but this is a great general rule to know that organics must have carbon and hydrogen. They can have some other things. Inorganics don't have both. Okay, and that's the best clear, simple, clear-cut rule I can give you. Okay. Now let's go into this. Let's see if you guys can do C5H5N5O. Now it sounds like I'm giving a chant here. Now C5 carbon, okay, but it also has hydrogen, so it's organic. C8H10N4O2, my favorite drug. No, not crystal meth. This is caffeine. My favorite drug, which I'm a little bit jazzed on right now, if you can't tell. I just happen to have a large iced latte just to get me going through my midday slump. I was feeling low, so now I am riding high on some caffeination. Carbon dioxide, CO2. It has carbon, but doesn't have hydrogen, so this makes this, guys, an inorganic. Sodium hydroxide, NaOH, inorganic. A protein called titin, C169,723, H270,464, N45,688, O52,243, and sulfur 912. Titin, organic. All right. So let's see if we can do some other things here. Okay. Um, so you can try to understand them by their molecule name, by a formula, by a structural formula, by ball and stick or space filling model. There we go. Let's get this word out correct. And try to understand the way they are shaped and get an idea. Usually you think of them as being like tetrahedral in structure or something along that line. Okay, or planar or tetrahedral, something like that. Now, what I want to think as well is carbon and its covalent bonds. Now, carbon is tetravalent. What does that mean? Carbon has 4 valence electrons. That valence shell is the second shell that wants 8 electrons. It won't 8, it only has 4, so it needs 4 more electrons. That means it will form 4 bonds, okay? So it has what we call tetravalent. Tetravalence allows it to form a complex set of structures. Other tetravalent molecules may be a good byproduct basis to build off molecules for life, like silicon. Now, carbon can also form things like they can be different lengths, like ethane, which is 2 carbons, or propane, which is 3 carbons. Butane, which is 4 carbons, okay? So we can vary in length of the hydrocarbon chains, or the carbon chains. It can branch. You can have butane, which is called butane because, as we can see here with butane, I have butane is 1, 2, 3, 4 carbons. Here I have 2-methylpropane. Now, propane is a 3-carbon chain. How do we name this? You go, now don't have to know this. This is for those organic chemistry students. You have the longest chain that you have is here. 1, 2, 3. You have one substituent, a methyl group. So we go 1, 2, 3. Then we try to make this the lowest number possible. So really it doesn't matter 1, 2 or 1, 2. So we call it 2-methylpropane. 2-methylpropane. Or sometimes called isobutane. Now we also can have double bonds in different places. We can have a double bond located right here. We can have a double bond located here. It can be now butene because butane is a 1, 2, 3, 4 hydrocarbon. It is on the first carbon, second the double bond. So here it's on the 1 second carbon, 2-butene. There can be rings like cyclohexane or benzene rings. So we can have all these little things there, okay? Now, hydrocarbons. What is a hydrocarbon? You heard me say this word. A hydrocarbon is an organic compound where it is only carbon and hydrogen. Okay, like methane, CH4, or C2H6. Okay, these are hydrocarbons. Hydrocarbons are organic compounds who are made up only of carbon and hydrogen. We call them hydrocarbons. For example, hydrocarbons are a major composition to fat. Right here we have some of the things that make up a fat right here, but we have hydrocarbons. Okay, the hydrocarbons, the black balls being carbons, and the white balls here being hydrogens. Now, naming hydrocarbons, I only make you memorize the first 4. This will come in handy as you move into classes, but the first 4 you'll need to memorize for my class. That's it. This is something I do that others don't do, but this is one of the things I do that some people don't do, but you'll need it if you had to take organic, and you'll be glad. And then some of the other ones that you get, butane, you start getting into pentane, hexane, heptane, octane, nonane, dodecane, things like that. And you'll start getting into those molecules as you try to learn the hydrocarbon numbers. So I'm going to give you guys a little bit of a mnemonic. Is MEP-B, MEP-B, M-E-P-B, MEP, M-E-P-B, MEP-B, 1, 2, 3, 4. MEP-B, 1, 2, 3, 4. That is the number of carbons. We name them based on the number of carbon atoms they have. Methane, MEP, methane, 1 carbon. MEP-B, MEP, methane, M-E-P-B, ethane, E, 2 carbons. Propane, 3 carbons. Butane, 4 carbons. MEP B, 1, 2, 3, 4. Okay? It's an easy way to remember it. Okay? And these 4 I'll expect you to memorize. These are the only 4 that I do in this course. There are others, but I picked 4. Okay? I picked the first 4. And you'll be glad I did if you have to take organic. It'll just be one more little thing you have a heads up that somebody else doesn't have. Okay? Now, isomers. Isomers, let's say I gave every student in class the exact same set of Legos. And I say, guys, have fun building Legos. And you go out and you build Legos. And every one of you builds something out of those Legos. But I say there is only one catch. You have to use all the pieces. Now, you build whatever you want. Use all the pieces. Okay? So one student goes and they build something like a car. Maybe the next student builds an animal like a horse. Maybe the next person builds a house. Now, each of these 3 people built something different. They use all the parts, the same parts, but they put them together differently. That's what isomers are. Isomers are chemicals that have the same chemical formula, the same chemical parts, but are not put together in the same way. Different structure. Same parts, different structure. Iso equal mer part. They have equal parts. They have the same parts, we just arrange them differently. For example, here we have the exact same chemical formulas. If I were to look at these 2 molecules right here, and I were to actually analyze them, we would see the same number of carbons and hydrogens in both of these. But one is called pentane, one is called 2-methylbutane. Why? It has to do with the how we name molecules. Now, here we have the same constituents. It's just that we have one on one. We have these little X's on the same side as the double bond. Here they switch. Here we have some same constituents, but again, some different handedness. Now, these are all isomers. Structural isomers, we just arrange the covalent bonds differently. Arranging covalent bonds differently. For example, pentane. 1, 2, 3, 4, 5. Penta, 5. A pentane is a hydrocarbon that has only single bonds and 5 carbons. 2-methylbutane, 1, 2. Okay. 1, 2, 3. We wouldn't want to, there's a methyl. We would want to go from left to right because this makes this be 2 and not 1, 2, 3. You always want to make this substituent be the lowest number possible. So it is called 2-methylbutane. All right. Now, what do we do, guys? These are the same chemical formulas, just different arrangement of the covalent bond. We stick this, we break this off here, bloop, and stick it up over here, okay? And stick a hydrogen on the end so we can put it back to where we, like how we had it. Now, what happens is we also have what's called geometric isomers or cis-trans isomers. Now, cis and trans. Now, I'm not trying to be offensive here or anything like that. I'm not trying to. This is not. This has nothing to do with that. But I am going to say this is the way we're used to hearing these words being used is in reference to gender. Okay, like somebody who is cisgendered, they they identify with the gender of their biological sex. Where if you're transgendered, you identify with the gender that's different from your biological sex. Cisgender, cis isomer. Okay, so cis and trans are words we're used to hearing with gender. This is different, but somewhat the same. Same use of the same prefix. Cis is the 2 little groups. We call these variable groups. You could use R or X or whatever you want to stick on there, whatever you want to call it. If they're on the same side of the double bond, they are called a cis isomer. If it switches to the other side, we call these trans isomers. Now, for example, there is a cis retinol. At the 11th carbon, there is 11 cis retinol. When light hits it, it becomes 11 trans retinol and it straightens out, which is in your eyes. This is the going from 11 cis to 11 trans retinol is the mechanism that first allows the process of vision in your eye. So enantiomers. Now, why do we have hands? Hands are non-superimposable mirror images. Now, I can't show this on a camera, but if you put your two hands together, your thumb is on your thumb. If you put palm facing each other and you put palms to palms, they are mirror images. It's just like you put your hand up to a mirror. But then if you look at the palm of both hands facing palms at you, your left hand has the palm sticking to the left, your right hand has the thumb sticking to the right, left hand has thumb sticking to the left, right hand has thumb sticking to the right. And then what you do is try to put the right hand on top of the left hand. That doesn't happen. The thumb sticks out on one side and they're not the same. They're non-superimposable mirror images. If you try to put this hand and pick it up, the thumb of that hand will stick out this way. And the thumb at that hand that way. They are, now, if you put them palm to palm, they are mirror images, but they're non-superimposable mirror images. This N2 here, this is a mirror image. If you try to take this and stick it over on top of that, that N2 would be over here, non-superimposable mirror images of each other. Okay? They're called enantiomers. Enantiomers can be very impactful. For example, one enantiomer, like the enantiomer of thalidomide, is effective at treating morning sickness. The other one causes damage to a fetus and birth defects. Now, there's a really bad thing. If you don't separate the enantiomer, one's perfectly good for treating somebody having morning sickness, but if you're pregnant, having morning sickness means you're pregnant, you're in that first trimester, and you're very likely to harm the fetus. And the wrong enantiomer messes up the baby. We always call them L and D isomers, left and what we call L and D or RNS. L and D and RNS are used for different classes of molecules, like amino acids, something like that, or some other molecule could be used. In this case, this is appears to be an amino acid. L and D amino acids. Which one is it that has methyl as the R group? I just forgot. Is amino acid that has methyl as an R group. Gosh, it's been too long since biochemistry. So I don't remember the structure of every amino acid ever now, when I had to know all of them. So anyway, I'll have to look it up. So here we have some examples. Ibuprofen, which is given as a non-steroidal anti-inflammatory, reduces inflammation and pain. The effective one is the S, which is the left-handed version of it, called S or Sinestra, and the R one, that is the right-handed, or REIT. Now, S and R are used in things other than like amino acids and some of your carbohydrates, things like that. A few of the organic compounds, some of them use that. Now, in this case, what we have is S ibuprofen versus R ibuprofen isn't effective. Only S is. Now, albuterol sulfate, which is a molecule that is used as a bronchodilator for asthma patient, only R albuterol is effective, not S. Why you pay the high price sometimes is for the chemist to separate out and actually try to make sure that only one isomer goes out. Okay. Now, functional groups. These are going to be big. These are what actually determine the functions. They are going to be what participates in the chemical reactions and give us the functions to the basic zincs. They have a function. Functional groups have a function. Okay, to borrow a phrase from my organic chemistry professor, functional groups have a function. And they're very important from the biochemical reactions. They usually participate in that reaction. Okay, now, case in point, here we have estradiol, the most important estrogen in a female. Testosterone, the most important sex hormone for males. Now, look at the differences. In estradiol, there is an extra hydrogen on the female. One hydrogen gone, so that covalent bond ends up becoming a double bond here, stealing away a double bond in this ring. There's a methyl group bounded here, which takes away that double bond. So when men sometimes say that women are too unstable, I point them right here and say, look at that. 3 double bonds right here and only 1 double bond right there. Who's unstable? Just saying. Okay. I'm funny when I'm a little bit more caffeinated, aren't I? Okay. So this will be the last thing we do, and this is purely memorization. You will have to go through these. I will hit about 3 of these on the exam, so you definitely need to know them. A few things I always ask is what they might be involved in, what they might be in, so methyl group, CH3, they are on methylated compounds. Hydroxyl, OH, are in alcohols. Carbonyl, which is carbon double bonded to oxygen, they can be found in amino acids and organic acids, especially aldehydes and ketones. Aldehydes have 1 organic group on the carbonyl. Ketones have 2 organic groups on the carbonyl. Carboxyls are COOH. You have a carbonyl, basically, with an OH bonded to the carbon. These are weak acids. They are found in associated organic acids. Carbonyls with an OH are found on organic acids like vinegar and things like that. Amino groups found in amino acids are NH2s. They are weak bases. Phosphate, PO4 2- on any phosphorylated compounds, any compounds forming phosphate. And sulfhydryls, SH groups, sulfur-containing groups. They are commonly found in things that stink, things that really smell. For us, we're very sensitive to sulfhydryls. Now, I'm going to tell you guys, it just takes time to go through these. You'll need to be able to go through these. I will say the functional groups are a good place for flashcards. When I learned them, one of the things I also did I took Post-it notes and I wrote them down on it, and I stuck them up like at my coffee maker, where I brushed my teeth and shaved in the morning. Places where I would be hanging out a lot, like if I would be sitting near like where my toaster and my coffee pot was, I would be there kind of heating up, or my microwave, I would be there making my coffee in the morning and heating up my breakfast in the morning. And so I would be there and I could stare at them and study them. And flashcards work good if you use them actively. You need to know their basic chemical formula, and if some of the things have them, which sulfhydryls I don't have anything on there about them. We just talk about sulfur-containing SH groups. There's a lot of things, like, for example, thiols, things like that, which we don't really discuss thiols ever. There are some amino acids that have sulfhydryls in it, cysteine, things like that. So what I want to discuss is know the basic formula and what has them. And I do need a little tweaking on this slide. I see a little bit here and there that just needs to be neatened up, made to look a little bit neater and prettier. As I go through my slides, I always go, I really wanted to make that better. I wanted to make that better. I wanted to make that better. All right, guys, that is it. And you can see these diagrams here pretty well. Hydroxyl, OH, carbonyl, carbon double bond to oxygen, carboxyl, COOH, a carbonyl with an OH. Here you can actually see this is acetone. That is a ketone. Propanol, that is an aldehyde. Amino group, NH2. Sulfhydryl group, as I said, cysteine. Thiol is a thing we sometimes call amines. And I probably need to kind of add some compound names and just really kind of go in and really try to be a little bit better on this slide right here, just to kind of match this a little bit better. Now, here they have OPO3 2-, and I just do this. I say PO4 2-. And then methyl group, CH3. Like 5-methylcytosine is an important molecule. This is a methylation of 5-methylcytosine is a very well-known mechanism to change and adjust gene expression. Really cool stuff. Recommend you check, if you are looking at a paper idea, epigenetics is a great one. 5-methylcytosine is something to look at when you are studying that, okay? 5-methylcytosine. That's an interesting one, and that relates to our methyl group, which relates right here. It's a good topic for a paper to do. Remember, that paper's due right before you guys go on your Thanksgiving break. Let's see if you can identify these. Number 1, carbon. Now, real quick. Corners are carbons. That's a carbon right here. Okay, there will be a carbon in each corner that you see. Corners are carbons. So what is this? If you said that this is a carbonyl, then you're right. CH3. CH3, number 2. What is that? If you said methyl group, you're right. Now, what I'm going to do is allow you to kind of pause it, by the way. Okay, I'm going to say these. OH, pause it, see if you know it. If you said that it was a hydroxyl, then you're right. Here, remember, carbons are corners. COOH. Go ahead and pause it, see if you know it. If you said this is a carboxyl group, then you're right. Okay, number 3, NH2. NH2. If you said it's an amino group, then you're right. Remember to pause. SH. Pause and see if you know what it is. If you said sulfhydryl, then you're right. Okay, COOH. COOH. This is a what? If you said that this is a carboxyl, then you're correct. Okay, what kind of isomer is this? Looking at it, I see a line. I see 2 isomers that if I pick this up and try to drag it over top, it wouldn't be able to superimpose. So I see 2 non-superimposable mirror images of each other. Pause it and see if you know the answer. If you said enantiomere, then you are right. What are these? Both of these things have the same chemical formula, but different structure. Pause it and see if you know. If you said structural isomer, then you're right. And this one here, I see a double bond, and I see methyl groups on both sides of the double bond. Then I see methyl groups on each side of the double bond. Pause it and see if you know the answer. If you said that they are cis-trans isomers, then you're right. This is the cis, this is the trans isomer because of the double bond position. Okay? These methyl groups related to the double bond. Yes, we're done, guys. That is it. Pretty short chapter, easy to get. The hardest part, I think, are the functional groups. Is memorizing them. That's it. Okay, guys, thank you so much. I hope you enjoyed this video, and I hope you enjoyed the lecture. This is the last one before your exam 1. Check your emails for instructions on that exam 1 in the next week. Thank you. Thank you, guys, and we'll see you in the next one.